My name is Kevin Chee. I'm a consultant advisor for Dr. Elaine Ingham's Soul Food Web. My background, just to kind of preface who I am, I've been on my homestead for about 7 years. It's a 10-acre ranch. I started farming back in 2019 and 2020, doing hemp production for license in the state for 2 years. In 2020, after kind of struggling a bunch for my first year, I decided to get certified in cannabis cultivation through Green Flower. I worked with Colorado Hemp Institute on some product formulation for a brief period with them. I started studying soil food web classes in the fall of 2021, and then I finished my final project for the consultant training program in the fall of 2023. I started consulting full time. Well, started doing consultant work at the beginning of this year and throughout this growing season. My experience mainly on our property is a lot of annuals in our vegetable garden and also fruit trees, some perennials, aspen trees, willows, etc. Um, but due to growing in Colorado and growing hemp for those 2 years, I do have a lot of experience on indoor growing as well. So grow lights, environments, understanding uh, light intensities, those kinds of things as well. I also, on our homestead, built in 2020 a grid-tied solar panel system with a good buddy of mine, and that pretty much takes care of all of our electrical usage as well as all of my devices that I use for indoor growing as well. So today's topic is soil bacteria, how the smallest living creatures create the biggest impact. And just to preface, like I said before, this is a short, about 15-minute presentation scratching the surface of soil bacteria. It takes many hours of studying to really understand what we know thus far about soil bacteria. We are always making new discoveries through the soil food web, through other universities and institutions about what they can do. It seems like it's a constant learning experience, even with the most seasoned PhDs of what bacteria can do for us as humans. So first, we're going to talk about the main types of soil bacteria. We typically will see cocci. These are circular or oval-shaped bacteria, most commonly and most associated in singular form in healthy soil systems. Bacilli are rod-shaped bacteria. These are also seen in soil systems, but are typically recognized with most dairy products like lactobacillus. Um, the other ones that we typically, not typically, but we sometimes will see, and usually common with anaerobic systems, are either coiled or spiral. These are be like spirochetes or spirillum. So when we see a single cocci in a system, that's usually associated with a healthy aerobic system, which all of our root systems are all aerobic. When we start to get into anaerobic conditions, that's when most of those bacteria will cause issues because the production of alcohol starts to proliferate. And alcohol, if many of you didn't know, will liquefy your roots. When we see chained cocci, so when we see more than one cocci or a duplococci, two cocci together, that can be another form of bacteria that we see in our soil. You could also recognize that in like the human mouth. Whenever we have a chain of cocci together, a streptococcus or strep throat can be an indication of some disease-causing bacteria. There are also affiliates of bacilli that are singled, but are also disease causers, like tetanus. Tetanus is a bacilli. The kinds of disease causers that we see in this spectrum for coiled and spiral are typically like your Salmonella, Shigella, E. coli, those kinds of things. And pretty much every association that we see this kind of bacteria, especially in large clusters, you have to be pretty mindful and careful about that particular sample or soil or whatever you're working with because these could make you sick, especially when we're starting to see ranges of the facultative anaerobes, which is 4 to 6 parts per million of oxygen. So recognizing the different types of bacteria is part of our ability to identify what's in our soil, the conditions of soil formulations, problematic areas, and our progression in changing a system from anaerobic to aerobic. All right, so what are the main functions of bacteria? We essentially have four. There are other subcategories of these as well, but I kind of broke it down as simply as I could. First is decomposition. Bacteria in healthy soil systems feed on organic matter and parent soil materials like sand, silt, and clay, extracting nutrients into its own body for heterotrophic replication, which occurs as fast as 5 minutes for that one bacterium to split into two. Another is soil structure. A healthy bacteria system creates alkaline glues that hold and aggregate organic matter and particles to create better structure for root systems and soil inhabitants. They also will bind together those particles to kind of aggregate more nutrients for plants as well. Nutrient cycling. Bacteria are based life forms for all of our predatory soil organisms that prey on the bacteria population, and that releases free mineralized nutrition right at the roots of your plants. So when a bacterium floating around and a protozoa or a nematode comes and eats it, some of that nutrition is absorbed into that predator's body, and then the rest is released as waste, but that waste is mineralized and ready for take up by the plant's root system. And then finally, a waste management. So bacteria are used a lot in composting facilities. They're used in the breaking down of organic matter. They're responsible for a large reduction of waste. There are types of archaea that decompose. There are types of archaea that decompose oil and gasoline. Anaerobes are capable of creating some of the most acidic compounds to decompose almost any kind of material, including ones that are radioactive. Bacterial biomass. So there are specialized soil bacteria that create nitrogen and nitrate, NO3 negative, and nitrite, NO2 forms. These forms of bacteria typically like pH ranges above 7. The biofilms produced by bacteria help maintain that pH level. Aerobic bacteria increase soil pH as they replicate and alkaline glues are formed. Specialized bacteria like Cellumonas carry cellulose-breaking enzymes that will decompose the hardy woody materials like tree bark. So bacteria typically will create more alkaline conditions if they're higher in numbers than the other lower life form, which is fungi. Bacteria are our primary method to heating our thermophilic piles of compost. As bacteria replicates using nitrogen-rich organic matter, water, and oxygen, they replicate at an amazing rate. One single bacteria can split in cellular replication every 5 minutes. This is how we create a pile of compost, and within 12 to 24 hours, our temperatures can reach upwards of 170 degrees Fahrenheit or 76 degrees Celsius. Allowing a full 200-gallon size of pile of compost to fully decompose and homogenize in as little as 3 to 11 days. So when I typically make my compost piles using the soil food web method, I'm usually done with that pile of compost and bringing it into a maturation phase, usually under 2 weeks. So bacteria are food for the predators. As bacteria are consumed by the upper levels of the predation cycle and soluble nutrition are released from the bodies of the predatory species of nematodes, protozoa, and others like rotifers and microarthropods. When the bacteria is consumed, parts of the mineralized nutrients is inside of the bacteria are held within the predator, and the rest is released as waste and taken up by the nearby plant. So you can kind of see here in this predation cycle that the base life forms, the fungi and bacteria, they get eaten up by the predators. Those predators are taking in nutrition for their body, but they also expel that waste. Everything is right near the roots of the plants because that plant is excreting those exudates, as Dr. Lane likes to say, the cakes and cookies, the sugars, carbohydrates, and that's what keeps the relationship of the bacteria near those roots. And then the predators will come along and consume those lower life forms, and they release that nutrition right at the roots of your plants. How am I doing for time? Doing all right. Okay. So the FTB ratio, our fungi to bacteria, is the relation of how we know the lower life forms and their ratios in accordance to the type of plant that we're trying to grow. So the amounts of bacteria in relation to the fungi will dictate what kind of plant succession will grow best in that particular soil. So for example, weeds grow best in a bacterial dominant or early successional type soil. Plants that are in the row crop section are typically anywhere between a 0.3 to a 1-to-1 bacteria to fungal ratio. Fungi create ammonium types of nitrogen, while bacteria can create the nitrate and nitrite forms of nitrogen. This is how the balance of organic forms of ammonium nitrates are formed in nature. And so when we have areas like the exposed rocks, lichens, and early successional grasses, you're going to see mostly a bacterial dominant soil type. So you're going to see, you hopefully want to see predators still in that system. In most cases, whenever I'm taking samples of somebody's soil, I almost always just see just an overload of bacteria, very little predators, and a lot of nitrifying bacteria only present in that soil. Excuse me. So when we move from the early successional phase to the row crops and going into the things that we normally plant in our gardens, that's getting toward that 1-to-1 ratio of fungi to bacteria. So we want to see the ratio between the two lower life forms start to just slowly become equal at that point. And when we get into the young forest all the way to the climactic forest and old forest growth, we start to see not a reduction in bacteria, just a huge increase in fungi. And as that happens, that soil typically starts to get more acidic, probably between that 6-to-7 ratio, as opposed to the early successional. You usually see 7 and above for the ratios of bacteria. All right. So healthy soils will contain large amounts of diverse kinds of bacteria as well as fungi, whose ratios will determine the health of the intended plants. This diversity will also include a large and diverse group of predators that consume those lower life forms and release the excess nutrients right in the rhizosphere. As Dr. Elaine has said, reestablishing the proper biology is critical. And then the biggest takeaway is that bacteria is only part of the larger picture. Having a healthy soil is combining all of the players in the soil food web. That was a brief description of essentially what bacteria does in our soil. Wanted to switch over and kind of go into questions, Stefano, if we can.
Yeah. I guess who here has got questions on bacteria?
I did have a couple of questions that were asked early that I can kind of go over first prior to those as well. Sarah B had talked about, is it possible to cultivate good soil bacteria for indoor plants? And if so, what are some of the good ways of going about that? When it comes to indoor plants, they can also benefit from essentially really good compost. The indoor versus outdoor are going to be using the same lower life forms and microbes in their soil. And so whatever you're cultivating for outdoor, you can also cultivate for indoor in your indoor plants. Good bacteria is pretty much anything aerobic for the most part. When we start to see a large increase of soil bacteria in any soil system, what we want to do is we want to introduce the predators in that system to start consuming them. And what we notice when we do our soil tests is that those numbers will start to drop as the predators come in. But as they start to drop, there's more soluble nutrition in that soil system as they're starting to be consumed. So when you cultivate, the easiest way to cultivate good bacteria for indoor plants, in my opinion, is having a really good worm bin to get those plants started. Here's another one from Yvonne S. How is there a way to know if you have good or bad bacteria based on what you're seeing on the plant at the surface? How would you eliminate the bad bacteria before it kills what's there? What would be your best solution to the bacterial issue that is seen rather on leaves, branches, or trunks of trees? So anytime that we see bad bacteria, it's usually a result of the conditions, whether that's low oxygen levels, overwatering, or uncomposted materials like manures getting onto those types of surfaces. So it's mainly not eliminating bad bacteria. What it would really be more about would be um, creating conditions that are allowing the good types of bacteria that we see to proliferate more. Outcompete is how Dr. Elaine likes to explain it. So we were trying to do is make conditions ripe for that all the good guys that are going to bring as much nutrition to our plants, whether that's on the surface of a plant or under the root system, to grow it in as high as population as possible and to cycle those nutrients as much as possible. So the times that we're actually adding those microbes and inoculums to the surface of a plant or tree or fruit trees is typically in the form of a compost tea. And we mainly use compost teas just for that particular procedure, is because when you're doing the compost tea process, the bacteria are replicating like crazy in that solution, and they're creating alkaline glues, which inherently makes that liquid very sticky. And so when we're applying it with a sprayer to the trees, usually Dr. Elaine has two rules for that. Usually before the first true leaves sign, or at the first signs of buds on your fruit trees. And so we spray usually in the spring, the beginning of the season, and that creates what she calls a castle wall or a barrier from disease and pests over the season. And so we're not necessarily trying to kill bacteria, we're trying to outcompete with good guys. Taylor G says, what kind of processes can you use to turn clay into rich soil for growing veggies? Kind of like the video mentioned with Lance, especially here I'm also in Colorado as well. So what we're trying to do with any type of soil is we're trying to turn, whether it's a sand, silt, or clay, into a loam. And the main component missing from most of those parent materials is organic matter. Most systems that are devoid of being able to grow plants into them is usually because we're usually in that 1 to possibly 3% of organic material inside our soil. So if you ever get a soil sample done of your soil, you're going to see all the different elements that an agronomist is going to list. That's part of the makeup of your soil. And then usually there's a small section where you see how much organic matter is in there. Most systems that are really difficult to grow in have very little organic matter. Now, if you're in a clay soil system, but you're upwards of 8% of organic matter, you're going to do really well growing plants in that type of system. So it's whether it's sand, silt, or clay, it's not necessarily about turning the clay or changing the clay, but just making sure to add as much organic material as possible, which is why also I agree in the HHA, they're talking about adding the cardboard, adding the mulch layer, getting that organic material on top of bare soil, if it is bare soil, and bringing as much organic material or composted material back into that system. And that helps what creates that balance of nutrient absorption, creating your levels of EC so that your amount of nutrients can be held in there, as well as water absorption and drainage as well, which is pretty crucial with soil systems that are clay, because clay likes to overload on water. And in most cases, it ends up getting stuck in a compacted layer and doesn't typically drain very well. So when you get organic matter mixed into there, those microbes are going to help break those compacted layers of soil so that you can get those root systems deeper, not just shallow. Savannah Y says, what is the first sign of dying or deteriorating soil? An easy way to tell is usually by smell. So if it rains really hard and you go outside and there's pockets or puddles of standing water and it smells, that's a really good sign that you've got really bad soil for the most part. Other ways is that if you see plants don't want to grow in a certain area, or you see an area that's devoid of bugs, if it doesn't have bugs and subsequently doesn't have birds and doesn't have other wildlife in an area, that is not good soil. And so either it's overly compacted, it's had too much tillage, too much heavy compacted machinery on it, a lot of different things that can create compaction layers in soil. If you see most of the life leaving that area, other than maybe flies, that's not good soil. You want to, again, you want to add conditions that are going to break up those layers of compaction. You want to add that organic matter in to help not just soak up that water, but help release it, dry it out. Dr. Lane does talk about using minimal amounts of inputs to condition soil systems if it's not doing it on its own. I use gypsum in building my swales in my heavily compacted, over-farmed land that I built, and now I'm building my own food forest in. And I think that's how Stefano found me, was on my swale video. Um, Other than that, yeah, I would say if you're really not using or getting versed in using a microscope, would be smells and then how other animals are reacting to that area. If a dog goes through an area, dogs are pretty keen on their sense of smell, and if they sniff a puddle and then they walk away, they're not drinking out of there because they smell something that's not good for them for a reason. Horses do the same thing. And so you want to kind of pay attention to what areas animals are avoiding and areas that animals are gravitating toward. And then lastly,
Jamie
Jamie
had a
V's.
question for you. She's
Okay.
uh, what kind of predators can be added to the soil to accelerate the cycle,
So
including
what we try to do is if you're not versed in using soil food web methods, you can add organic matter, and that organic matter is going to contain the nematodes, the protozoa, the diverse predators in the system that you can add to it. There is products that you can do for the indoor market, like predatory nematodes that you can buy and add it to certain things. So you can kind of make your own blends of vermicompost by adding certain things to it, different kinds of compost, organic matter. Um, and if you get really curious with a microscope, you can kind of figure out how to cultivate your own predators into a system. But the easiest way is adding that organic matter in there. Because if you're adding fresh leaves or grass clippings or even weeds for that matter, and just kind of chopping things up, and that's going into either a cold bin or a thermophilic bin or a vermicompost bin, you are already adding more diverse types of microbes, including predatory species, especially if you get it from multiple different areas. Some from really lush areas, from not so lush areas, some from treed areas, from those edge lines that we talk about in the HHA. So meadows, forests, shrub lines, anything that you can get from multiple different places and add and aggregate together is a great way to get predators in the system.
Kevin, most people are smaller, like and home gardeners. What do you think the best way? Just get like a worm farm you could do yourself. That's probably the most simplest way, right, to get high quality soil without a microscope and analyzing it.
Yeah. So even if you try to make a compost pile, which when I first finished the first stage of Soul Food Web training, which is the FC course, I did my own composting based on what that part of the beginner part taught me, and it didn't exactly go the way I wanted to. I kind of screwed it up. I was luckily able to reach out to a consultant who graduated back in 2014 and and he kind of advised me of certain things. He's actually one of the top cannabis consultants in California from the Soil Food Web. He's done, I think, a million square feet of cultivation over his career, and basically started coming up with something. But he says you can always change it into vermicompost. So I took my partially finished, partially unfinished compost and put it into a worm bin, added worms, and this is by far probably the most congruent conversation I've had with most other consultants and graduates of the program is that you can even take the most perfect compost and make it into worm compost, and it becomes better. Worms will do continual refining of all that organic material. Even if it starts coarse, by the time they've gone through it, it will eventually start to become fine. They will continually break down that system, and it's not very not a high amount of maintenance to do that. Occasionally checking in on them, if it gets a little drier in a dry area, sprinkling some water on top, maybe putting a layer of carbon on top to make sure that they stay nice and moist. But I would say that was probably the biggest area of my accelerated success for getting through the consultant training program and the lab tech program was basically having a bin that I could always go back to and take my samples from and always be looking at healthy protozoa, healthy bacteria, healthy nematodes, and just being able to constantly have a resource that I know is going to grow plants well because it's got all of the right types of microbe diversity.
Awesome. Thanks. Diana had a question.
Yes, thank you. I seem to be getting real problems with, first of all, the damp in the UK and in the compost pile, which I think is really a bit too small. So it doesn't seem to be hitting the heat. So I think probably what you're saying about taking the compost through another system is probably going to be the answer for me too. But I seem to be getting a huge imbalance in that I lift the lid and it's just swarming with wood lice and slugs. So this is in an enclosed plastic bin with a lid, and I think I'm exacerbating the whole problem here by having it in this plastic container that's just sort of sweating and holding the moisture in. So I just feel like I'm taking that compost, then putting it into my raised beds, and I'm actually transferring the problem into and underneath my plants, which then, even if I net or contain or cover, they seem to be eaten to shreds. And I think, well, I'm actually importing it into the beds because it's probably full of eggs and all sorts of things. So can you just confirm if my idea is a right suggestion or I need to tackle something else?
And you're saying this container that you have is your worm bin?
It's a cold compost bin. I
Okay.
don't think it's getting anywhere near, for the size, it's only barely a meter by a meter by a meter.
Okay.
So I think because of the damp, it's just not heating up enough. So
Well,
basically, it's not like killing off maybe the eggs that need to be killed with the heat.
yeah, when we're doing a thermophilic compost, we do a minimum of 200 gallons, which is um,
Mm,
in feet, it's 3 feet by 3 feet. It's basically a large cylinder, a small
right.
version of like a Johnson suit almost, but we're doing thermophilic. And
Okay.
essentially, what we're trying to do is we're trying to get our temperatures to continue to raise. We get above 131 degrees Fahrenheit
Yeah.
for a period of about 3 days, up
Yeah.
to 170 degrees Fahrenheit, and we're essentially killing all the eggs, the weed seeds, and then at that temperature, we actually are killing off a percentage of the disease as well. And that's kind of a partial sterilization of what we're doing in that thermophilic phase. And so if you're doing cold compost, you want to make sure that the compost is still generating some heat. If
Yeah.
it's not generating enough heat at all, or it's not steaming, even if it is wet, there's not enough nitrogen in the pile for the bacteria to replicate and create that thermophilic phase of heating. So when
Okay.
bacteria have enough nitrogen, they
Mm,
they it's the law of thermodynamics. They essentially will heat up because they're consuming a bunch of energy and then they're splitting and turning into another one. All that activity creates all the increase in temperature. And so,
okay.
if you don't have enough nitrogen, you won't get the heat. And if you notice that your pile just never really changes composition from the thick pieces into more of a granular type, then
Yeah.
the components of the pile aren't quite correct.
Okay.
You can take everything together into a plastic bin, but you want to make sure you still have good drainage so you're not getting anaerobic and then it's not just holding, slugs and the things that could potentially eat your plants inside of there. You want to make sure that you have air in the top, and then the one that Stefano has listed as the prize, that's the same one that I have. And there's air in the top and air in the bottom. So you want to make sure
Okay.
that whole system stays aerobic, because worms are obligate aerobes as well.
Yeah, I can see there's a whole series of problems with the bin, so maybe I'll just get rid of the bin.
Or you could modify it, you could add more air to it, you could make sure that it's maybe elevated above a certain amount so that any excess water is allowed to drain, so that it's
That's a
not
good.
allowing those slugs and those things to, because if it's really wet, that's perfect environment for slugs, because their
Mhm,
bodies are very wet and mostly water. And so
mhm.
worms in that case will start to outcompete, because worms will eat other things other than just the organic matter. They'll predatorily they will eat other consume other bugs as well. And
Okay.
so and then I've noticed, there'll be spiders that get into my bin and things like that, and they're eating the other arthropods in the bin as well. And so the types of bugs that aren't going to harm your plants are the ones that you want to see in your bin. If you see the ones that are possibly going to harm your plants, you definitely don't want to add that to your garden beds. You want to wait until your structure of what you're trying to build looks beneficial, at least from the bugs that you can see.
Right. So the $1 million question, what predates the slugs then?
Slugs and snails.
Yes.
Oh, I got the answer for this one. Duck,
Have you?
you have a duck deficiency. As in permaculture, you're missing ducks in your system.
I can confirm.
Oh, okay. I am missing ducks.
Yeah, you're duck deficient. I'm sorry.
Yeah,
Yeah.
your soil food web has a long chain to it. And typically, and we can go into maybe into depths of this, but Dr. Tom Dykstra does talk about plants in general when their Brix levels, which is essentially their carbohydrate and sugar production levels, is the best way to measure how productive a plant is. When you can
Mm,
take a couple of leaves, and what I recommend is using a garlic masher and just a simple refractometer is the easiest way to just check it. They're pretty cheap and they don't cost a lot of money. And you can kind of start to predict what your plants will be defended against. And anything below a 9 in total sugar production, your plant
right.
is going to have some susceptibility to certain types of bugs. Between the 9 and the 12 range,
Uh-huh.
grasshoppers are no longer an issue on a plant. And that is because grasshoppers can't digest complete proteins. They can only digest incomplete proteins. So when you have certain pests on your plants, it's because that plant's not producing those secondary and tertiary metabolites in order to create better cell structure. And that's
Uh-huh.
the whole purpose why nature has pests, is because they're meant to essentially decompose and kill all of the stuff that's dying or dead. And so
Yeah.
when you have a whole bunch of different kinds of pests, it means that your Brix level or the health of your plant is not at
Hmm.
its full potential. Anything
Hmm.
above a 12 in Brix is objectively healthy, and slugs, snails, grasshoppers, any other insect is not going to want to come near it. They may take a bite and then they're just going to leave, because that
Okay.
plant is producing enough of a healthy immune system that those pests aren't going to enjoy eating on that plant. They're going to just move along.
Right. That's great. So really, it's a case of get more air in the bin, get it heating up, so maybe make it bigger. And I wonder, I might add some more wrigglers to it as well.
Yeah.
Would
Really
help.
good decomposers. Yep.
Okay. I think I'll try that, see how I get on. That's brilliant. Thank you so much.
You're welcome.
Hey, Kevin, on that point, so grasshoppers ate the crap out of my plants this year in my garden. I had plenty of nitrogen. My corn was 10, 12 feet tall. So to get the breaks up, could I just spray like molasses on the plants? Would that work to get the
That's only going to increase your bacterial counts. That's not necessarily going to so what I'd recommend is if you can make a solid compost pile or a vermicompost pile and let that age over this winter time, and then make a tea, that your tea will include the molasses. Your tea will include typically things like fish hydrolysate or a humic acid to make the fungi grow as well, because you don't want just bacteria. You want the fungal component as well. That fungi will also create that barrier that we talked about to spray on your plants as they're starting to grow, because we don't have microbes. I mean, everybody has microbes everywhere. There are microbes under the soil, but there's also, we want microbes on the surface of the plants as well, everything above ground. And so if you're going to spray that tea, get it onto the surface of the plant, and then create that defensive mechanism early. If your soil's still going through that conditional phase of getting to as best as it can be, if you're still working on it, I would suggest the tea in the beginning of the season, as well as what we call a compost extract. So we take our good compost, we put it in a 400 micron bag, and then whether that's a gallon, 30 gallons, 250 gallons, we massage it. I use an air pump using a GOT brewer when I'm doing large scale applications. Like I was doing some disease mitigation for 4 acres on commercial property to reduce. They had a ring fungus, necrotic ring fungus, that was basically these dead rings all over all their grasslands. And so the extract will flow with water into as deep as the roots go, or as deep as the water will go. So the difference between an extract and a tea is that tea is meant to be sprayed because it's sticky inherently. The extract will flow with water, and it's a lot easier to make, and it's probably the easiest remedy you can do to act as not only a soil conditioner, but an additional fertilizer and an inoculum to get all your microbes into the soil. So you take your compost, you put it into a bag, you add it with water. I usually let it sit for a few minutes in the water, get somewhat acclimated, and then I start massaging the bag, and that releases all of those, not just organic material, nutrition, but also microbes into the water. And then I can take that and either apply it with a watering can into my garden beds. I can put it into a giant sprayer, and I try to do as much passive spraying as possible, gravity fed type drip systems usually. Because when you run it through a pump, you will lose a percentage of your microbes as it passes through the impeller. Hopefully, you can use a system that doesn't use a heavy impeller or a very slow moving one in order to move the microbes out of a tank onto the ground. But there's multiple ways that you can act as a defensive system. When I got my plants into the field to do my final project, I had 28 tomato plants in the field. I had 14 on one side as my control and 14 on the other as my treatment. When the grasshoppers came in July, which they sometimes will do when you get a wet spring in Colorado and then a really hot June, July, and they're just everywhere. And I have this documented, which was part of my presentation for graduation, was that all of the plants had grasshoppers on them. All of them did. But I was only able to find one plant that had a bite out of a tomato on the treatment side, as opposed to probably 75% of the control side had damage from grasshoppers almost all over. The fruit from the that had gotten damaged started to rot on the control side, and I lost probably 3 or 4 plants on the control side. The treatment side, they were on the plants, and I would walk through and they'd jump off, but I could only find one tomato out of, hundreds that actually had damage on it. And over the course of time, it actually healed itself up, never diseased the plant, and the plant continued to flourish. And the rest of the entire rows, I ended up with 25% higher in growth on the treatment side versus the control side by just doing extract. And I did, I believe, 6 applications over the whole season. And by that time, I think I had done like 4 applications up until that July point when the grasshoppers just exploded all over the field. And essentially, they didn't touch everything on the treatment side. I did my Brix measurements between the two sides, and they both looked healthy. They both were producing tomatoes. My control side was at a 7% Brix. My treatment side was at a 9.5% Brix. And the treatment side had zero, essentially zero damage. I mean, maybe a percent or two, but that was pretty much it. And we had an amazing harvest just on our treatment side. And I didn't net the plants. I didn't do anything else with the plants to try to cover them from the grasshoppers. But I objectively saw in real time that Brix levels does have an enormous effect on the ability of the plant itself to produce what, in my opinion, was higher pheromone levels and defensive levels that the the pests were just like, no, I'll take a bite, but no thanks. No, I'm moving on.
Wow, that's awesome. That's a great story there to share. Anybody else have questions for Kevin?
I didn't have a question. It's just I guess a bit of a comment on for Diana with the bin she's got. So here in the UK as well. So again, quite wet, slugs all over. And I think what I found is just having a variety of things. So not relying only on that one bin. Because if you're having compost bin with worms and everything, for me, it didn't work the first time. So now I have got a worm bin where I put only food scraps. Then I have got another bin where I put all of the big trimmings out of the whole allotment. So any big branches or anything like that, I'm putting in there and I'm rotating them. And they're quite large, made out with pallets. And what I do once a year, I rotate, mix it all up, move it onto the other side, and you just carry on, keep moving it. Because big piles will just carry on decomposing super slow. You're not going to get the heat in there unless you're actually constantly on top of it. If you follow, the guy, what's his name?
Oh,
here
yes.
in the UK.
Yeah.
Forgot his name, but
Charles Dowd.
Charles Dowding. He's got amazing compost, but the amount of time and space they've got available allows them to achieve that temperature. If you're doing it at a small scale, I have got two big boxes of pallets, two of them on one side. I've got one of those plastic bins. And that plastic bin, what I do is when we have a lot of windfall, all of the fruit or any waste food from the allotment that we've not been able to deal with, I put in there. Then I've got separate the warm bin. And then I also have one of those rotating bins that is supposed to be like a hot compost bin that you spin like a tumble dryer.
Tumbler.
In that one, I also put all of weeds. So anything that I don't want to be propagating, I put in there, and then that's composting on one side. So you end up doing composting, but in lots of different versions.
Yeah.
When the big piles are not decomposing very quick, then I can grab sections of that and put it on the vermicompost on the worms. But then I can sift it and then take partial bits of those and then put it on the hot bin with the rotten fruit. So you end up doing several cycles of that composting from different bins at different timings. I don't know. On my end, all I'm doing is I don't want to waste anything, but I'm going to give it as much time as it needs to actually decompose correctly, so that when I use it, it'll be good quality.
Exactly. It's no point, is there, really making something that's a non-effective product and then spreading it all over your vegetables.
And you'll see it. Well, I don't know how often it happens in the US, but in here, how often people will be throwing away all of their waste. They'll have their garden bin and then you'll be chucking it away. So I do gardens on the side, and then I encourage people to keep all of the trimmings. Everything that comes out of the garden stays in the garden. Let's find a place where we can actually put it all in there, whether you want to compost or not. Let's just have a place where you put in everything in there, because eventually it will turn into nothing. And if you're lucky and you're organized, then you'll be able to get something out of it. But
yeah, I agree. It seems like I'm going to be spending a lot of time in my compost bin area. Thank you so much, George.
The other thing I did as well is hedgehogs. They are fantastic for slugs. So then having lots of habitats around there will help you quite often. And then avoiding as many dark places where they can actually hide underneath. So unfortunately, if it, like with me, it's an allotment, I'm going to have them. So I just then focus on what I can do and just accept that there will be slug damage.
Yeah. That's all good tips. Thank you so much.
Diana, please share with us on the group. Take pictures of your compost pile so we can all
I will.
And the progress. That'd be awesome.
The before and the after, you mean? The success.
Yeah, exactly. Let's do it.
Okay. That's like a challenge. You've thrown me the challenge now, haven't you?
Yeah. The gauntlet has been passed. So. Sweet.
Brilliant. Thank you.
Guys, any more questions specific? Kevin is going to do a bunch more of these soil food web. The next presentation will probably be fungal, I assume, nematodes. So every time we're learning a little bit more Kevin also teaches soil food web. I put a link on the Perma Resilience course to his course if you want to learn more. Kevin, if you could just talk a little bit about your course and like what people will learn, how to use a microscope, how does that work?
Yeah, we make it as I would say rudimentary as we can. The biggest two biggest things that we learn in Soul Food Web is getting really good with knowing how to use a microscope and identifying the amount of creatures that we have under our slides. And then the second is compost building. So that's essentially Dr. Lane makes us expert with the microscope under the looking at soil samples and then doing all the different kinds of compost, primarily thermophilic. And so, because our remediations are sometimes quick, I know that you can change a system from a really dead or barren system into a thriving one with animals and do that over the course of decades. But we try to actually change a system within a single season, so that plants are 200 to 300% productive in just that first one. But my program is meant to just kind of get the it's not a replacement for FC. It's probably 10% of what the FC program is because it's quite expensive to go through the Soul Food Web programs. So this is mainly just a taste of it, and then I'm also starting to get you into the practice of doing some of the Soul Food Web stuff. If you're interested in getting to FC, so it's kind of it's a bite of getting into the practices of Soul Food Web and kind of what was my specific specialty, which was really understanding the microscope, how to use it, and how to really dictate what our samples are looking like, and just kind of crunching through those. I actually have a couple of colleagues that are lab techs that work with me on this course as well, and they're there to answer questions and kind of help people along.
And one thing I want to emphasize to group and everybody listening to this recording is like there's only, I think, 18 certified level of what Kevin is for the entire program. I did the soil food web, and very few people get to the level Kevin is at. So I highly encourage you, bring pictures of your garden. We're obviously not in the gardening season for most people, but bring pictures of your garden, anything soil, take pictures, bring it to the next chat Kevin is in, and then you get personalized consultant looking at your soil from that regard.
And we do have some lab techs, so I don't know if they're on the call, but they had said that also if you guys are looking for the biological assessments of what you need for your samples, and I'm not sure some of the international ones, we could probably hook you up with who's available in other countries. I want to say there's less than 50 worldwide consultants, but there are more lab techs getting into the program and getting certified around the world. So if you need help with that, to kind of go through those things, either you want to do the program or you just want to get an assessment on your soil and to kind of look at what microbes are there, we can try to help with that as well.
Okay, sweet. Awesome, Kevin. Thank you. Anybody have any other questions before we go?
I did see one more question from Jamie that said, what is the best way to inoculate biochar with beneficial bacteria or beneficial microbes? Simply mixing with compost or compost tea. Dr. Lane does talk about biochar to a degree. From her studies, biochar may help with certain more mineral-based types of amendments to soil, things that I'm talking about from just the chemical makeup, not necessarily for microbes. A lot of people think that, well, you have biochar, the microbes are gonna inhibit the biochar and stay there. The microbes stay on everything. They aggregate pieces of organic matter together, and they'll stay wherever they feel like staying. So biochar does take a certain amount of effort to make. And so if you add it to compost, things like that, it's just another step that you have to take. If you're trying to minimize steps, you don't have to add the biochar. It's not going to make a huge difference whether you do or don't in your populations of microbes.
Thank you, guys. Thank you for this, Kevin. Appreciate you.
Cool.
And I think we'll do one in every 4 weeks if that's all right with you.
Cool.
And we'll get the next one going on the next soil food web. So awesome. Thank you, guys.
Awesome.
Thank you, everybody.
Thank you
Thanks,
so
Kevin.
much.
Thanks, everyone.
Thank you. Bye.
Bye-bye.